So welcome everyone. So as Michael pointed out, the talk is about some sort of a merger of trying to understand how we can use biotechnology in a rather interactive fashion. And what this kind of picture depicts here is, right, like we have a human, we see something microscopically, like cells under microscope, and we actually can see this for a couple of centuries now, like look through a microscope and see what's going on there. But what really has eluded us is to actually manipulate these organisms or kind of play with them. And of course, like if you're in a research lab, you may be able to do it, but kind of lay people, even like in a school setting, it's usually passive observation. It's not kind of true interaction. And this was kind of an inspiration for me like a couple of years ago, like just this thought, like could we play games with these sorts of cells? And this kind of led to many things even well beyond anything that is intended to be game or playful. So let's start here to kind of also give credit to this audience. So what's the difference between these two pictures? And what's maybe also common? Just some thoughts.
One's a lot smaller.
Okay, one is smaller. What else?
The age and training.
Age, yes. Maybe the training needs, yeah.
They both have interaction with technology in some way.
Okay. Other things? Right. So in both cases, you see humans interacting with a computer. There's about 50 years difference in time. You see that this machine here is much about number crunching, occasionally breaks down, and it's by far less powerful than what you see here, where even children can have a very interactive experience. And so you can say what you see on the left is number crunching, what you see on the right is kind of an interactive experience. And if you now think about recent developments in the life science or biotechnology, we can actually say there are many parallels what's going on now in biotech versus what has been going on in computer science or electronics a couple of decades ago. And just to make this a little bit specific, so what you see here is two versions of a so-called microfluidic device as an example. So here on the simple one, you see basically two inlets, the white and the blue fluid, and they kind of merge and you see how droplets form and how these droplets kind of move around, right? And much more complicated versions of that you see here, which is kind of like an integrated electronic circuit, but instead of moving electrons around, you move small fluid volumes. Imagine, for example, you have a droplet of blood, put it in a device like this, and then the device is capable of doing certain diagnostics for you, counting a number of blood cells, running certain assays, and so forth. And these devices, as I said, get used for diagnostics. They get better and better in terms of their capabilities. Used a lot in research, but have not yet hit, so unfortunately here it's kind of not showing in the back, have not yet really entered the consumer market, but they're kind of similarities in the way these things scale, right? And so this kind of prompted this thought, if we have now these kind of technologies, when do they become interactive, consumer-friendly, in the same way as electronics become, and if they do so, what could we do with them, right? And just as a general side note, like lots of things that go on in bioengineering, we can really look back at kind of computer science and being inspired, and certain things we can do similarly, other things work out completely different. And so for me, the inspiration of that was the first one, electronics and computer science gave rise to games. Why aren't we making games in the life sciences? And the idea is what are then called biotic games, is like, could we make games where a human can interact with a biological process or material, and this really has something to do with modern biotechnology. So if it's a pure emulation or pure simulation, that's not what I mean. And I also don't mean something which is macroscopic like horse polo, which is maybe more ancient biotechnology, right? So that's not what we're interested in. We're really thinking about something that is kind of microscopic and relying more on the modern technology. And what you see here is kind of one of our first projects that kind of shows the paradigm as such. What you just saw is a single-celled organism, Paramecia. This is like a very simple microfluidic chip, about 1 centimeter wide, and here you see some electrodes, and if you apply mild electric fields, then actually these organisms start following these field lines. And you can now bring this whole thing in real time on a computer screen, and then, as you see in this inset, kind of push buttons and then move these cells around and play games in the sense, for example, you have a virtually overlaid object, and the task is to kind of make the cells move through this object, right? You could either see this as just an interactive experiment, or you could see this as a game, okay? And so this is kind of an example of what interactivity means. So If you now just change the skin of this, right, like putting other objects on top of it and kind of changing the logic, then you can make other games, so to speak, right? Like, so you have an underlying game engine, if you want to say. But in that case, the game engine is something real. It's real cells, okay? So here again, the device, this is kind of rather simple, right? Like this microfluidic chip, like a simple webcam, and then just brought on a regular computer, and then putting some abstract objects on top of this. So what could we now do with that, and where should one go? One inspiration for all of this is kind of education. I think lots of the initial applications of our work will be in this realm, and then we'll also talk later about more serious type, I mean, education is serious, but kind of about other applications that are beyond particular education. So one thing that always fascinated me, and from a life science perspective also made me a little bit envious, if you look at other fields, they really have cool educational toys that are very attractive, right? Specifically in mechatronics, like kids building these cool robots, and kind of these robotics competitions around that are really supported by NSF and others to really engage children, students in kind of learning these technologies. And in the CS realm, more recently, there's equivalent challenges here, like the National Video Game Challenge, with the idea, hey kids, program games, it's a fun activity, but thereby you become engaged with programming, which is an important skill to have, right? And so could we take these kind of things, these kind of ideas basically into life science realm with our kind of interactive setups. And so this brings me to a second project where we used, try to develop a similar system as I showed you before, but used organisms that are a little bit more sturdy, that just respond to light, and developed a simple microscope where you can actually play these games on a phone, and also having the idea that you can actually build the phone or kind of the whole setup yourself and thereby learn something about device design. So the organisms here are called Euglena. These are single-celled organisms about 50 micrometer in length, so that's half the size of a human hair. They're green, so they do photosynthesis. They have this red eye spot where they kind of sense light. So basically they can just look in one direction. And then they have this long flagellum, which kind of makes them propel and swirl through the fluid, okay? And it's actually the same structure that, for example, sperm tail has, or also kind of cilia in your lung, so very conserved structure in biology. And so we made a system where you can now play with these cells on a phone. So here you see these kind of cells in action, right? If you follow more closely, you see kind of their swirling motion. You may see that they actually roll around their own axis, right? You may also see, if you follow some of these particles here, these are passive particles that they undergo Brownian motion, but at the same time, kind of the fluid field moves around. Who has heard about low Reynolds number hydrodynamics? Anyone? Right? So the thing is, if you think about water and swimming, we have this macroscopic experience that we have, right, like with vortices and all these things. If you kind of scale down water in these kind of length scales, for these organisms, it feels like swimming in honey. So for example, mass and inertia doesn't play any role. As soon as they stop swirling, they instantly stop, and then all that drives them is diffusion, if at all, right? So very different world to live in, and very fascinating world, and also something to it's worthwhile to kind of bring to lay people, to everyone, right? So to give you a little bit of an impression how we think from a biophysics perspective, how an organism like this works, right? Here's again like this Euglena with this one flagellum, and you can basically abstract this thing as an object which has this flagellum and basically can undergo not all 6 degrees of motion, but primarily kind of drive forward, roll around its own axis, and then kind of execute this yaw and pitch motion. You have this eye spot, which is kind of reading light from one direction. Now you can actually ask yourself questions like, how could you make a feedback loop between these axes and the light sensor to make something move very robustly towards the light or away from the light, things like that, right? But this kind of behavior underlies then these setups. So here's the setup in real. So this is 3D printed microscopes. Actually, as we were just discussing before, one of the undergrad classes here at Stanford, students built something like this in order to learn a number of subject matters. Basically constructing a device. You have a little microfluidic chamber here. again, where these organisms live in. We have 4 LEDs from 4 sides, so by actuating each of them, you can drive these cells around. And then there's some simple optics that basically puts it on the phone. And then we can then make games with that, similar as you've seen before, but now a little bit advanced. First of all, it's colored now, right? So the organisms on color. But then we also track individual cells, right? You see how an individual cell is basically tracked and also amplified bigger on this side. And then the game involves basically like using the joystick to shining light from different directions in order to orient the cell or make the cell swim towards this virtual goal. You can also actually tab on the screen and select the cell. So there's kind of this playful aspect, but then there's also this thing like, I mean, can we basically thereby emphasize something interesting and teach something about biology? And we can, because there's kind of scale bar here, right? You see something about size. You see kind of the instantaneous velocity of the cell, which is currently tracked, right? You see how fast these cells move. You can also see something about kind of in this close-up behavior, right? Another game we kind of implemented is kind of a Pac Man version, okay? We kind of try to somehow make this cell to swim along that sort of path, and if you follow it for a while, you see it's actually not that easy, right? It's not, which brings to an interesting point. It's not that you really control a cell like this, but it's more like you control a stimulus, and then you kind of observe how these cells behave. You learn a lot about kind of, I don't know, biological noise and variability and other things as well. And actually, at the end of this particular game, you kind of even get left with kind of a trace of that you followed, which may be interesting to see how well you did, but also at the same time contains lots of interesting biophysics about these organisms, right? To build this particular thing, this is the whole building plan up there. So it's really intended to be affordable or accessible with very simple means. You have a simple electronic circuit, some sticker microfluidics that you can put it together, 3D printed microscopes, and very simple eyepiece. And so idea is really that, I don't know, in makerspaces or whatever, even in middle school, children could do this, but really, I mean, learning about these very different subject matters in a very integrated manner. And if you talk about the Mechatronics that we had before, right, which is basically like this on the top and here, so we be more inclusive about different matters. Just to say, one can also do some more serious activity, like go away from these games and just, for example, track cells, just do directed experiments and then formulate hypotheses. Does a cell, for example, on average move faster when the light is on or off, right? People generally assume it moves faster when the light is on because it tries to escape the light, but that's usually not what one finds. We also wanted to integrate something more from a modeling perspective, and so for those of you who know, it's kind of a children programming language, Scratch, and so you can actually animate cells that behave like the real ones in an abstracted sense, and this is actually the whole code you need to make a cell like this move. So this is kind of the early stages. We have shown this to a number of teachers, and we kind of now hope to somehow get this into a classroom and actually being able to refine it and seeing how this really works or could be employed. Here's some user studies, so some children using it. This from some science fairs, like different children of different ages drawing what they see. Kind of funny enough, like this 8-year-old kind of drawing little legs, kind of realizing this is kind of a live organism, although they don't have legs, right? What you generally find is that about 10, 11 years of age, kind of children grasp the concept, which is also kind of the age where children would use it in school, actually, kind of where the biology lessons kick in accordingly. We also ask a number of teachers, have them come to our lab and ask them about different aspects of what they think they would find useful about a game setup like this. And most of them felt like the building aspect and kind of being able to kind of select individual cells and making measurements is maybe the most useful. The idea about playing games and maybe modeling, I mean, teachers still express enthusiasm, but kind of seem to be of less interest. And so, as I said, we're kind of now trying to get this into classrooms and seeing how this would work. And just to kind of summarize this part, with this particular setup, we think we have a system where you can really learn on 3 different scales, right? You can make the thing, you can build it yourself, you can play with it, kind of playfully explore, but you also can make more quantitative measurements and do kind of more serious science, like, I mean, how fast do these cells move and how fast do they respond to light and other things, okay? So another project using the same cells was now the idea, how could we bring this to museum space, but also how could we get a more intuitive interface even. You may have noticed or not, like when I explained where the LEDs are coming from and how the light is, maybe some of you were confused and also some of the people are confused while playing it. Yes?
Yeah, sorry, this is about the previous stuff.
Okay.
So have you compared it to other more traditional teaching methods?
No, so I mean, we taught Stanford undergrad course where I let the students do these kind of projects. So that's kind of one set of things. And then we engaged with teachers and had a few kids try it, but we have not even reached the stage where I said like it has employed in a few classrooms and it worked on itself. Like the comparative things is even a level beyond that. We want to do, but it's a little bit out. Yeah, so basically making a more intuitive interface even. So what you see here is again the same cells, and this is a touch screen, and you see these cells swimming, and you can draw on the touch screen, whatever you draw is actually projected onto these cells. And as they kind of avoid light, right, so they're again kind of in this quasi 2D chamber, as they avoid light, you can kind of capture them, right? So just to rerun it, so just watch this one cell coming from above. The user is drawing this kind of light, and the cell kind of bounces back, right? And so this is actually very intuitive because what you do, you see directly, right? So here's kind of again capturing that sort of motion, where now the cell basically bounces around in this virtual light circle. So the setup itself is kind of shown here. It's essentially a normal. You can even use a pocket projector. Exchange the optics, and instead of projecting large, you project small, and then you have a standard microscope with everything, right? And put back on the touch screen and establish this feedback loop. It's also important and interesting if you have also an eyepiece that people can actually look into the microscope itself and see what they are doing. What is shown here is the same setup, but now a different magnification, right? Like what you can do easily with optics is just zoom out. And also slightly different light intensity values. And here, for example, you can also see how these cells, after some time, start accumulating in certain colored regions. Again, kind of avoiding the blue light. Right? Yeah. So this is the setup. Here's kind of this packed projector, some optics. Here's a reservoir where we actually keep the cells and kind of automatically flush them. And so that's flush them into this chamber, which is here, which is also important. A whole question about long-term stability, right? Like we are very used from our computers. We can turn them on and off, and this works for years. So with these systems, we know that on a couple of weeks time scale, the system works well, and then we have to exchange all the organisms. In part, I mean, when I say exchange, we have to exchange them there, but we exchange them much more frequently, every couple of hours or so inside the chamber, right? But if you kind of want to enable these kind of interactive experiences, we also need to make sure that we deliver the user something that is robust, right? And it's kind of an ongoing thing. And yeah, and in this thing, you actually see the eyepiece. If you look through the eyepiece, what you see in there is the cells swimming around, but you also see kind of the light that is projected, right? Okay, so just some other things you can do with this. For example, you can do experiments, like let the user choose different colors and ask them like how do they respond to different colors. There's of course also a question like how do we set overall intensities, but we made it such that they respond to blue light but not to the other ones, right? And so users can get that sort of experience recognizing the blue color. You can also do an experiment which is a little bit more challenging in trying to draw over the front or the back of the cell and kind of realizing where's the eye spot. It's challenging in the sense due to the lags in the whole system, right? The projector has lag, the touchscreen and so forth. It's very hard actually to capture the back of the cell, but if you draw over the back of the cell does not respond. It only responds if you draw over the top, over the front of it where the eye spot sits, right? So this kind of allows you even to do experiments where you do subcellular resolution interrogation. So this is how we had tested it a couple of times in the San Jose Tech Museum, where essentially you have the touchscreen here and then the setup next to it. People can actually look inside, see the whole equipment, and then we also have this eyepiece to look through. And we essentially left this kind of with two signs. We're saying like, look here or draw here, and then we were watching from apart, kind of doing user studies and also post-interviews, like what would people do, how would they interact with that. And just to show you some examples, so usually people come up, just start drawing something, as is That's what you do if you encounter a touch screen. This is like a 4-year-old, for example, right? Like people writing their names. And then what they often realize after some time is that there's actually stuff moving in the back, and there's actually the stuff that is moving is somehow interacting with what you have drawn, right? And then people start playing more. The other aspect is usually one person is occupying the touchscreen, the other person has nothing to do, then starts looking through the eyepiece and suddenly realizing that what the other person is drawing is coming in the eyepiece, and then people start having conversations with each other. So here's an example of one user doing kind of an experiment, kind of self-activated play or experimentation. The whole frame here sequence is a little bit over 2 minutes since this person has walked up to the setup. And what you can see is initially the person kind of captured a cell and then made this little maze and tried to see how this thing escapes the maze, right? And so what I would argue is that we enabled a person to making a microfluidic chip within 2 minutes, right? Without ever having done anything on that kind of microscopic scale, right? Yeah, and also mentioned before, like this importance, like what you draw on the screen, you see through the eyepiece, and then people kind of invented games like this where, like, yeah, draw something, I guess what you draw, and the other way around. So from a more broader context about museum experiences, if you haven't been to the Exploratorium or the Tech Museum, if you go into kind of physics space or so, like you have lots of like pendulums and other things you can interact with and play, and really get prolonged engagement where people spend like a couple of minutes, parents with their kids. And in biology, this is actually historically much harder to achieve. I mean, you have like kind of macroscopic petting zoos, but the microscopic world is usually through a non-interactive microscope, or if it's interactive, then it's more about controlling the microscope, but not interacting with the actual living matter. And so I think we can really, through these activities, change that. And here, let me just show you another project which we started together in collaboration with the Exploratorium. I mean, it's the same setup as you've seen before, but now we use a Kinect to just record your whole body motion and just project this body motion in there, right? You see woman with a child dancing, and then you see on the bigger screen on the left kind of the projection with these cells. If you follow this one closely, and it's highly sped up, you see how the cells basically avoid that person, so to speak, right? And also this picture frame, how you can actually trap a few cells and move them around. And one thing I hadn't appreciated before we started this project is actually kind of like the organisms and the human kind of meet each other in their corresponding worlds, right? Like you see macroscopic yourself on the screen and the cells at your scale, but if you look through the microscope, you actually see a little person dancing around in there with these cells, and that's actually what kids like to see, right? Like, so, okay, so these are kind of interactive experiences for museum setting. This is kind of ongoing. So now, want to get some other ideas about yeah, like programmability of these systems and what could we do. And just want to say, I'm, from my personal history, when I was a teenager, kind of also inspired like when the home computer revolution came about and like kids having computers at home, primarily wanting them to play games, right? Parents thought like this is educational. And in the end it was, right? Like I, for example, learned programming through my computer that I wanted for gameplay. And so the question is, could we around the space create something similar where you, I don't know, have a device like this that you could program? What could you program? But then also maybe do some more serious thing. And so this is an ongoing project where I basically combined the two setups that I showed before, like the one with the projector and with the LEDs, which means now we can manipulate cells from like in a global field with this projector, but then also give some directional motion from the sides. And so here's an example what you now can do. This is much more zoomed out, right? These again, these Euglena cells, but very, very small. You have this light pattern where you basically construct some sort of barrier which the cells don't want to swim through, but then you also shine light from one side. And what you can do with something like this is, for example, concentrate cells, right? Like you shine the light from here, all the cells swim over there and get captured in that sort of chamber, right? And you can even call this this whole thing, what you see there, you can script this up and make a command out of it, concentrate cells. Like you call this command, and then this is what happens. You can do another command like this, which is kind of clear screen. What is it? Yeah, okay. So you basically do the inverse thing, right? Like shine the light from here and kind of get rid of all the cells that are in there, right? And so you have certain ways of manipulating, and now you could ask yourself, like, what's a exhaustive enough programming language that you want and what you could do with it. So for illustrative purposes, we basically programmed the game on this, which we call bugs and boxes, that allows a demonstration of these sort of capabilities. And the idea, so this is kind of the startup mode. The game hasn't started yet, but you kind of want to clear part of the screen and kind of move some cells. Now this is where the actual game starts, and this game has basically the goal to get these cells into here. The light is on from this direction, and what you as a player need to do is kind of get rid of this barrier, right? Like, and the player didn't do this, so he lost because all the cells accumulated here, but it did not move in there. Okay. And now what I'm going to show you a movie like of 3 levels of this that are all kind of successful. I don't know why the... No. Okay, so again, first level, cells need to go in there, so you do it successfully by erasing this boundary. The timer runs up, you have enough cells in there which are tracked, so good. Next level is don't get any cells in there, so basically you need to draw a boundary. You do this, keep all the cells out, very good. And then the third level is basically get the cells in here but not in here. And so again, the lights are turned on in a certain sequence, and you basically need to make a barrier here and erase there, and then all the cells end up in here but not in there, right? And so I actually have, just for better overview, kind of summarized this here, right? This kind of shows how we can really program levels, how we can program behaviors. And so again, the first level, the light is coming from the left. The player is asked to kind of get rid of this barrier. The end goal is to get the cells there. And the next one, the light comes from the opposite side. What you need to do is actually draw a boundary in order to prevent the cells from being there. And then the third level is more complicated because first this light is on and all cells move down, and then later the other light is coming on and all the cells move there, and you need to kind of do 2 operations, right? So also, to be honest, so this is something you can do in the lab space. It's not yet there that you, I don't know, can hand this off to someone and play this with as much enthusiasm, for example, as you do with a regular video game, which are highly polished, right? But I think what this should show is that these systems have a potential for writing programming languages about it, on it, and also doing higher level manipulation, which may inspire similar things like early home computers, like enthusiasts now kind of toying around with this, but may also have more serious applications if you kind of think of how we can manipulate kind of micro-robots or swarming robots in microscopic scales, which is also kind of an active area of research, right? Okay, so, and now let me come in the second half of my talk, kind of later half, more about this idea about cloud labs. So how can we actually now use these kind of systems to allow people to interact over the web, right? And the general idea is a student or researcher sitting at home and running experiments over the web, right? Where you need to have an actuator and a sensor. And the system is, again, very similar, basic setup as before, like 4 LEDs. This is kind of the website. You see the camera view, the live feed. And then you have this joystick here, this virtual joystick, which allows you to actuate individual LEDs. Here you actually see an external camera view onto the microscope where the chamber is. And if you follow it for a while, then you see, when the light is coming from here, that after some time, kind of cells start moving up, right? And you can basically playfully explore just by going to a website. And again, appreciate that these cells are not deterministic. There's all sorts of stuff going on. Image data is generally rich, both from which makes really room for discovery, both for kind of student level, but maybe also in the future for researchers. And here's like after the experiment, you can actually then download the data and written some scripts where then the system automatically kind of tracks the cells for you, presents all this kind of track data that you can, for example, import as a MATLAB and then doing analysis with that, right? So we did a number of user studies with this. One of them together with Paulo Blickstuen, who's here at the Stanford School of Education. So we went to a middle school in San Francisco, and the students basically were supposed to do things, kind of doing some basic measurements and observations, but on the other hand, making some modeling, and we kind of wanted to supply this. And the study was basically designed such that within a 55-minute class period, students would do some pre-tests, and it would spend like 5 to 10 minutes working on this platform, then doing some analysis of the data, and then we had some modeling equipment, some modeling tool where they could kind of try to fit a model to their data. First thing is, so in the pre-study, we tried that all the students do their own experiments, and that actually did not work so well. Part of the reason, kind of bandwidth limitation in a school setting, right? And then also we not having enough systems in parallel to supply all the students with. What we then actually decided to is that all the students kind of did an experiment together where this system or the web page was projected here, and then all the students said like, hey, let's see what happens if we put the joystick this way, and kind of there was this in-class conversation over like 5 to 10 minutes and doing this experiment together. So then the students in pairs did a data analysis. So basically then they all got access to this kind of movie that they had just generated, and we also generated some other pre-processed images where we stacked a few images, and it's kind of right away easier to see in which direction cells will move. And then students were discussing, do they move away from the light to the light, doing some basic counting statistics. And then the last thing is we provided them with this modeling interface where you have a pre-recorded movie here in the background, and you're supposed to make this thing here, this model, kind of match the swimming path of a real organism. And there's 3 parameters you can tune. One is the forward speed, one is how fast the organism spins around its own axis, and the third one, how strong it responds to the light. And if you kind of choose these 3 parameters correctly, then you get kind of a path that looks pretty similar like this, right? And then students kind of engage in discussions, and really to emphasize like these organisms just have this one eye spot, so they can only sense light from one direction. What they then really need to do is basically roll around the axis to kind of explore the 3D space. And that's also leads to these kind of undulating motions, as you can also see, like kind of this tumbling motion. And yeah, so the students were doing this and kind of discussing and basically comparing the experiment that they've done and the modeling side by side. If you kind of look of like kind of the overall outcome, students learned the content. We had like 40 students about. Also importantly, that the students kind of perceived themselves as successful. They enjoyed the activity and felt they learned something. And also the teacher's feedback was positive in the sense like that the students were more alert than on average during the average class time. And teacher also mentioned it was as a positive thing that all these logistics about setting up the biology, I mean, are kind of taken away, and that you can even in a much more compressed class time do experiment and kind of more sophisticated modeling bring this about. I also want to emphasize, I'm not saying all teaching biology should now run over the web, right? I think the hands-on is important as well, and also pure simulations have their place, but I think this could be a way to really deliver certain types of experiments that are currently not possible in school settings. And if you now think of like how could this really scale up in the future, so here's actually the setup or the setups in our labs. Each of these is kind of one of these microscopes, right? It's basically a webcam, these 4 LEDs, and then an objective. And then the idea is, I mean, we set up a whole system where you have multiple users that can basically run experiments and in parallel being routed to the best PPU at the time, setting up queues and all these kind of things you just need to build out to allow people at any given time to, access these sort of systems. And again, to emphasize, since we can auto-monitor the system, so from time to time the system itself runs a little experiments and checks whether the cells are still responding properly, and if not, kind of flushes fresh cells in, and if this one fails, then we have to exchange it. But by doing this, we can really have a system that runs autonomously and then also really scales from a technical point of view. And yeah, it just was mentioned this here. You kind of see how 3 different of these systems are running, right? And you see, for example, in this one, there are not many cells in there. So the system would automatically recognize that and then change something to it, right? Really, having this auto monitoring framework in the back is important. So how does this now really scale from a cost level, and how useful is this? And I think this really can make a difference nationwide and also worldwide in terms of education. And just if you think about it, like these experimental systems are rather simple. I mean, setup maybe costs like $250 or so, including like a Raspberry Pi and a webcam to kind of operate them. And then if you kind of consider like these organisms are very simple and cheap, a certain maintenance cost of also maybe $250 a year, and assume that an experiment, usually as now experiment is a minute long, right? Then you come up that you can run about 500,000 experiments a year for much less than a cent per experiment, right? And any given systems. And if you now assume that, I don't know, you have 5 million students in the US every year taking a certain biology course, then you can see how with 10 to 100 of such systems you could really kind of make a difference in educational settings, okay? And really at kind of a price point that is low enough that would cost a couple of dollars to, for example, run a biology class, which is maybe even cheaper than kind of ordering the organisms to begin with, okay? And so future ideas are then, of course, also how to extend this, first of all, to other stimuli. So I think it would be great to kind of integrate that sort of system with this much more intuitive surface of kind of drawing colors over it. And what you see on the right is actually like an example of different organisms. So these are again Euglena, but then the big one is Volvox. It's a multi-cellular kind of colony. And this actually has, under these conditions, positive phototaxis. So it actually swims exactly the other way. And if you follow it closely, you actually also see how it generates certain flow fields and kind of makes all these cells that swim by actually move around, right? So there's some visions that in the long run we could really go to other organisms, other stimuli, electric, magnetic, whatever. Those things could be built out, right? Okay, now I want to tell you about another example of such a system which actually predates the one that I just talked about, which is very different in terms of scale, throughput, also stimuli, and which I think is important to show because also draws some lessons on like comparing the two of how one should actually design these sort of systems. And this Cloud Lab works with an organism called Fusarium. It's a slime mold. So what you see here is a petri dish, which is about this size. And in yellow, you see this organism. It's a multinucleated single-celled organism that kind of spreads around, forms these nice networks, and kind of grows in a dish. And these red dots that you see is animated where basically food was dropped by a robot remotely, right? So red is basically a trail of food that has been placed, and then you can see how the organism explores and then actually grows along the food trail. And so it's also an interesting organism in the sense that kind of development and behavior, right, are kind of tightly coupled, like the shape of this organism and how it moves are kind of the same thing. So just to note, the movie that you see here runs on a course of about a day, right? So basically the system takes an image every 10 minutes. And the system itself that we built, we prototyped out of Lego, so kind of pipetting robots, where on a flatbed scanner we had like 6 such dishes, and then the robot would kind of place these food drops based on user interactions or user-defined food droplets on there. And then we built also kind of a web interface out of it that you could, for example, run on a phone or via interface to do your experiments. And just to show you, so this is kind of the interface, right? Like you go to a website, you pick your experiment. This is a dish which is currently running, so to speak. And then you define your food trail or food stimulus by, for example, just drawing this out, right? And this then gets put in a database, and then on the back end you have these machines that now run these experiments, right? So this gantry robot is now taking up fluid, and then it's a little bit shaky. You see here these individual dishes. Now the scanner is taking a picture, and then these pictures are basically distributed to the individual user, and then you can come back and see what you've done, okay? So here again the setup, like the robot, this is kind of the pipetter. It's all Lego except kind of a simple syringe, and we can actually reliably dispense fluid like down to 10 microliter. And this is kind of a server rack which has 3 such machines, each of them running 6 experiments, which kind of was important for us to kind of see how these things scales and how you distribute many experiments over multiple machines. Each machine runs multiple experiments. From an architecture point of view, it's actually different than the Euglena system before, because the Euglena system was always like with this live system, one user checking out one system and then kind of operating directly on. While here you have a kind of an asynchronous mode where each machine is autonomously kind of checking once in a while in the database whether there's something to do, and the user is also interacting with the database, just putting the stimuli there, and looking at recent data, but the user never controls any system directly. And this actually has the advantage that if you now have setups here that run many experiments in parallel or run high throughput, that you can really distribute multiple people's kind of work over multiple setups, right? And if you think about modern biotechnology, what's going on there right now, there's lots of high throughput equipment out there, right? Where like you can run thousands of experiments in parallel, and so rather one scientist running thousands experiments, why not having thousands of people run one experiment each? So kind of making these couplings. Right? So you see some examples where the students did when we tested this in a course, kind of trying different things like how well this organism responds to food, where they can actually make the organism kind of split, right? Or what happens if you give the organism different sizes of food, like would the organism be able to kind of discriminate between that? And then there's some homework, kind of doing some fractal analysis, some modeling, and other things. So here, for example, is a series where a student made some observations of how organisms of different size, the shape actually depends, and then made basically a biophysical model that would kind of capture this. Important aspect of all these kind of online systems is also kind of the potential for doing learning analytics on this log data. And just to give you a glimpse of that for this particular system, so this is kind of the 10-week period over that the course ran. Each of these blocks is an individual experiment. Each of this red bar is when the student logged in, right? This was usually when the experiment was running, and this was then later looking back at the activity. And then also a student could actually look, which is shown in pink, at other people's data. And so it gives you the end of the quarter like a profile of how individual students, how many experiments they run, how often they logged in. And these are like 4 different students, right? And just looking at it, you see different profiles. For example, with this student here, the blue bars, which always go from the first time to the last time the experiment was done, are much shorter. So this is a student who usually did an experiment only based on the last experiment, but never looked much further back while the others did. We see a student on the top who looked at lots of other people's data, right? So we haven't really lifted what's all in this kind of data, but this has potential to really understand and track how do people do experiments, how do students learn, right? And this is, I guess, something that you guys are all very familiar with. Here's some simple robots which we now try to kind of also get into schools where you can do some simple experiments, doing concentration gradients with food colors, which is something that children like. And thereby, what I showed initially, kind of these robotics things, right? Like, it's not only about robots that kind of can move around, but it's also how could we make robots or inspire building robots and interacting with robots that do kind of life science research, right? Kind of to bring these two together. So the general utility for these kind of cloud experimentation setups, I mean, we talked a lot now about education, right? I think there's potential, and it's one of the next project, is like having a dedicated small set of real scientists interacting with a platform like this and seeing what would they discover, what kind of research would they do. We could hope that high throughput equipment is better utilized, and actually now some companies out there, that kind of cloud lab companies for pharmaceuticals and also academia to run experiments for you. And then there's this whole notion of citizen science, where, for example, Turner, which is also here, was developed here at Stanford, like allowing people to really contribute to the research by running their own experiments and analyzing, right? So if you kind of abstract from this a little bit, like what kind of equipment does it take to enable interactivity? We know, first of all, of course, it needs biology. It always needs a sensor, it needs an actuator, almost trivial. And so we now call this kind of a biotic processing unit, similar if you think about BPUs or CPUs or GPUs, right? And what would also be very useful to have each of these setups or these BPUs interacting through digital input and output channel, right? I mean, you can imagine making this interactivity work by pipetting with your hand or visual inspection, but if you really kind of follow this sort of paradigm, then you can really integrate this in any sort of existing framework, right? For example, a web interface. So yeah, so that's kind of the definition of a BPU, digital input output, and then you have biology in the middle that you can both measure and stimulate, right? And so you could kind of treat it as any sort of electronic device that you have. Performance metrics of such a device would be interesting, right? Like imagine you want to deliver education at scale for the US. How do you do this? Which kind of device should you choose? Which has the most interactivity going? And you could, of course, say, let's just see what's the throughput of the projector and the camera itself, right? And this is not very meaningful because if there's no biology in the middle, then there's no interactivity. And so the truth, what may be best, is kind of doing a mutual information measure, where you basically look at if you make a stimuli, how much of the stimuli can you read back from the response that you see, right? And just to illustrate this a little bit, so if we have, for example, two LEDs from the side, we have these Euglena cells, you apply a stimulus, and response time is on a scale of 10 seconds, you can basically have a mutual information like 1 bit per 10 seconds, kind of seeing that sort of stimulus. If you have 4 LEDs, you kind of double that, right? If you have this kind of projector system, which you maybe can tile up and have different people running on different parts of it, you maybe become more like 30 bits per 10 seconds. If on the other extreme we look at this Physarum system, right, which had a much lower throughput, we only got an image like every 10 minutes or whatever, you're way lower than that, right? And so I think having those sort of kind of coarse measure could really be informative of what to do in order to enable interactivity at scale and also cost. So some Outlook comments. There's a question about ethics here, which we sometimes get asked, especially when we make games out of these systems. Just to name a few of these objections, right, like animal welfare. Do we respect life when we play with them? Are we playing God? The yuck factor, so some people may say, oh, it crosses me out seeing these cells. There's a slippery slope, right? Nowadays we do it with cells, but do we later do it with monkeys? Trivial pursuit, right? Is this kind of a waste of taxpayers' money? Public safety, maybe Superbug evolves while we play. Game ethics, right? Are these games kind of well constructed and playful? What would happen if we do citizen science on this and someone makes a discovery, who owns it, right? So there are a bunch of things you can ask. And so together with David Magnus, wrote a piece a couple of years ago and kind of addressed these issues, thinking it's fine to do this kind of with this microscopic organism, given that we follow a few guidelines. First of all, no pain, no harm. We should engage with the public and saying what we do is good, right? For example, being educational, informing the public about modern biotechnology, respecting life. So, I mean, what does it mean to have a respectful interaction with a single-celled organism? But you can ask yourself. And of course, respect the player, right? Like whoever interacts with the system, you want to give something positive to them. Yeah, and then kind of really circling back to my intro slides, between comparing biotechnology and computing technology, right? You can say each of these fields went through kind of the early really foundational discoveries, like, for example, the quantum mechanics and the golden years, right? And then we had the transistor and the integrated circuit. And similar thing, you can see like the structure of DNA, DNA manipulation, and now these kind of circuits that we can now build out of microfluidics or kind of synthetic circuits. And if you want to say so, you can draw a correlation line and find some sort of a 50-year relationship between them. And if you look where games stand, right, it's always a debate what was the first video game, but you would kind of see that sort of a 50-year relationship. And then we can also ask, like, what was the time when devices really came into households and people were using it? And based on that, it should not be that far away. So it should happen in the next 5 to 10 years. If you really think about it now, about your phones, smartphones that you have with you, right, there's already certain bio measures in there about heart rates and other things. It won't be long that, I mean, already like if you have blood sugar level measurements, right, won't be long that these things have microfluidic devices integrated and that we then can play these kind of things on our phones. Maybe another way of getting these things in everyone's hands is kind of the cloud labs that I talked about, okay? So bright future ahead. And so let me thank my lab, which is over there. Half of them works on that, the other half works on more conventional life sciences, so to speak. And just to summarize, so I told you about interactive biotechnology. We have a number of platforms, and we spend a long time on kind of making them robust and freely programmable, and that's kind of an ongoing theme. We use them kind of in different sort of setups, right? Self-builder kit, museums, clouds. This concept of this biotic processing unit is something, an abstraction of a device that then basically everyone could use, even if that person wouldn't know anything about biology. And the applications now, I mean, talked a lot about education, but it's a question about research next and going into micro-robotics or what other kind of things could we manipulate actually on these sort of scales. Okay, and so if anyone here would like to interact with me, my lab, more collaborative way, a more formal, more informal way, always happy to talk, always interested to have good people. Also want to point out that this is Zahid, who's actually a CS student who made a brave jump from CS into bioengineering. I think he's doing very well and he's having fun and doing these kind of things. Okay, thank you very much. Questions? Yeah.
You showed me the examples when the cells were, the sensitivity was light and the motion was the flagellum. The question is, have you played with other things where you actually use light to open ion channels or use light to play with DNA and have different genes being expressed, and then use a language on top of it, like an interactive language, and start mixing things that you open ion channels, express genes, and put things together in multiple and different cells to make really complicated systems like in Lego?
Yes, so that would be the vision. We would love to do all of this. The thing is, we are happy to have a robust system now that just relies on a wild-type organism. We don't have to do any genetic manipulation or other things. Right now, I think let's focus primarily on this organism and run with this because it's stable. Any new organism, any new manipulation, I mean, there's labor, there's logistics, making it robust. But in the future, I'm sure it will come, right? Like many more types of interactions and organisms, biological materials will be used. Yeah.
When you were getting started, how long did it take you to find this creature how many others did you try?
So it was pretty fast. Like the very first game I showed you with this Paramecia. It was rather, I don't know, it took a couple of weeks and we were happy with that, but then it kind of hit a roadblock because they weren't stable enough long term, and it took them quite some soul searching and trying lots of different things until we ended up with this organism. The way it worked in the end, like I went to a school supply company, bought everything I got, put it all on my desk, and whatever had survived after months without doing anything, that's what I worked with. Right? And had a strong stimulus response, right? Like shining light on, you always see a response. Yeah.
Do you find these creatures in your local pond, or do you have to?
Yeah, so these Euglena, I mean, they're very commonly used in school education. You find them in your ponds. They're safe to eat. There are even companies that make Euglena cookies in I think, Japan. We can buy them. They're supposed to be very nutritious. I haven't tried them. Yeah.
And it seems like almost the future iteration of, the male home ant farm in some ways, or the caterpillar, the butterfly thing. And I'm wondering, in many ways, those were pitched as sort of you can watch it, and maybe there was some lesson like stages of the chrysalis and all these kinds of things. But I felt like it was more of just like a close-up experience. And I'm wondering is your point of view that you want that kind of thing? Or are you intending these more to supplement very specific, like pedagogical goals in science or biology education? So, for example, like what specific, if that's the direction, what's the specific kind of lesson that you want someone to draw out of these games around light sensitivity or flagellum? Like what are the concepts you're trying to teach?
Okay.
Or is it just sort of giving an intuition and access?
Okay, so first thing I'm very strong about is interactivity, right? I do something and I evoke a response. I can make those organisms do something, right? And the other thing is, these organisms stand as an example of many things you can learn, right? If you want to teach someone like how does microbiology work? I mean, you can pick one organism and study it in depth, and then you can show a picture about other organisms or show a movie, right? If you want to demonstrate how can you make measurements and statistics on a living system, I mean, you can use this. Or how do you make models? So I think they can really supplement many more things by just choosing this single one. Yeah. So there was a question over there? Yeah.
Oh, yeah. My question was kind of similar to Michael's, but yeah, along these lines of, I guess I'm wondering what you guys were doing when you had, or have you thought about other types of material that you can use in the classroom in terms of the types of relating that to these specific, learning goals or other things like that? Because it seems like so exciting to use this, and then I think there seems like there's a lot of these possibilities, but I'm wondering what are the limitations you see in like the specific organisms you're using now, or is it, I guess if you could speak a little bit to as you look to expand this, what are like the next steps in terms of, is it the organism that needs to change, or is it the input that needs to change, or how do you go, or just
If you want to make these things work, you need to have a few features. Like one is, you want to have responses that are rather fast, right? If you see something in 5 seconds, better than in an hour. You want to have something that's very stable and has low logistics in terms of keeping it alive or keeping it happy, right? And then you want to have some easy to use stimulus, right? Like light is easy to turn on and off. But if you have, for example, chemicals, right, like that you flow in, how do you get rid of the chemicals again? So I'm not sure whether I forgot something, but there are a few things that make it logistically extremely easy or make every project extremely hard. And so I would love to have more organisms, more stimuli and things like that in the future. But as I said before, right now, let's just primarily focus on this organism and let's go more demonstrate the applications, right? Like demonstrate that we can do a cloud lab at a scale and it's educationally effective. Yeah.
So I guess this is related to these last two questions, but what really are the pedagogical goals of the games? Is it just to interact with the creatures, or is there something deeper? And are you achieving those goals?
Okay, so I can tell you, I personally came from the game idea, and maybe in retrospect I should have thought about interaction and just games as a sub part of this rather than games. And so I kind of evolved along with that. Whether we meet these education goals, I mean, that's really the question. That's only something we get into, right? I mean, I showed you like this data from this study that we did in San Francisco Middle School, right? I mean, we see the children learn something, the children respond positively, the teachers have positive things to say, but this is basically starting, right? And whether a game itself, I mean, I believe in the pedagogical value of games in many aspects, or more general, just the capability of interacting, right? To playfully explore. I think this is very important. But we would need to show this still, right? And some of the teachers that I showed you earlier, some of the teachers were more skeptical whether the game is really the right way to go. Maybe the more standard biology experiment is just the important thing to do.
Another related question is, you could just have a simulation of these creatures, right? Okay. That would be a lot easier to deploy, that would be a lot easier to...
That's a very important point, which I maybe didn't make too well. So there are certain studies showing that if you tell people, like, this is alive, this is real, or this is simulated, that under certain conditions they respond with much more interest if they know it's alive. It was also in a museum we actually, where we had the setup next to it, we kind of also removed the setup, so they only saw the screen and then asked people, and then many people then didn't realize it was alive and they was much less interested in it, right? So simulations certainly have a place, and they are much better in augmenting certain things. They may be much cheaper to do things like that. But on certain conditions, having the live thing leads to much deeper engagement and more interest. Yeah.
Does your lab work with the other virtual reality labs on campus much? Seems like there could be some cool stuff.
Not yet really, right? I mean, no, not yet. But, I mean, none of if these systems get stable enough, it would be cool. I mean, that's what I was saying, this abstraction, right? Like, can we deliver a box like this BPU, which is just digital and the biology is living in there, but whoever, like in a virtual reality lab, can just treat this as any other electronic device, as any sort of physics engine. If we kind of can enable that, I mean, then I hope that people make interesting stuff with that, right?
Yeah, it seems like your main hunches are the interactivity and the fact that it's real. And so if you use virtual reality, like that's like kind of the combination that it makes, like interactivity in real life. It could be really cool. No question.
Yeah, good. Yeah, so we're kind of thinking, like, could we even animate, like, not animate, kind of, could you somehow swim through this space with the Oculus Rift? I would like to see that. I would like to do that.
All right, let's thank the speaker one more time.